Perfect. Start off with thank you very much to the organisers for inviting me here to beautiful sunny Barcelona to speak to you today. This is my first one of these meetings. I am a computational human geneticist and not an experimentalist, so bear with me on any of the experimental work as I get there. But today I'm going to take you through a bit of a journey that's been a journey of mine over the past kind of 18 months and talk to you about small nuclear RNAs. So most of us in this room and most people in the clinical genetics world focus almost exclusively on the around 18 to 20,000 genes in our genome that directly encode proteins. But there are even more genes, around 25,000 or over 25,000 that are annotated in humans to date, that are actually non-coding RNA genes, so they're not translated into protein. And so far, these genes are pretty much untapped for rare disease diagnosis and also for mutational scanning approaches. So I'm going to focus on one subtype of RNAs today, that are the small nuclear RNAs, or snRNAs. So snRNAs are critical components of the spliceosome. So this is this massive protein and RNA complex that mediates the removal of exons from the pre-messenger RNA and the splicing together of the exons in the final RNA transcript. Now, RNAs are, these small nuclear RNAs are critical to every part of this splicing process. They have these names like U1 and U2, where U1 recognizes the 5' splice site, U2 recognizes this branch point, and together they recruit this so-called tri-SNRP complex of U4, U5, and U6. And these together recognize the 5' splice site and catalyze that splicing reaction. And I'm going to focus on one of these very specifically, this U4 gene that I only became aware of in about February last year. And that was when this amazing student in my team, Yu Yang, came to me and said, I found this really interesting looking variant in the Genomics England 100,000 Genomes Project data set. Now, he'd not been looking for this. He'd been looking at potentially translated regions within non-coding RNAs. And this variant had been flagged as a high impact in our pipeline. But this variant is a single base insertion, and it's found in 46 different individuals in Genomics England, and 40 of these are known to occur de novo. That is, it's newly occurred in a child and doesn't exist in either of their parents. So I will admit, I said to Yu Yang, this is clearly a sequencing error, this is rubbish, please can we move on? He proved me nicely wrong and said, no, hang on a minute, I think there's something interesting here. Oh, sorry, I was supposed to flash this up. The reason why I thought this was wrong and more likely a sequencing error is these de novo variants are random occurrences of variants that happen in the germline, and we expect them to be relatively evenly distributed across the genome. So to find so many individuals with exactly the same variant is just really, really fishy. But Yu Yang, as I said, was persistent, and he said, hang on a minute, all of these individuals have as yet undiagnosed neurodevelopmental disorders, or NDDs. And this actually comprised about 0.5% of the just shy of 9,000 individuals with undiagnosed NDDs in the Genomics England 100,000 Genomes Project. We couldn't then find it anywhere else. So there are about 55,000 individuals in Genomics England that don't have NDDs, that didn't have this variant. And when we looked in the population cohorts, it wasn't in NOMAD or all of us, and it was in a single annoying individual in the UK Biobank. I still don't know why. My only assumption is that it's somewhat mosaic. But nevertheless, we get this stupid odds ratio for a rare disease of 2,500. So we started looking into this, thinking that, okay, maybe this is a little bit interesting. So this is what I'm showing you is the entire RNA. It's very short. It's 145 base pairs long. And this variant that Yu Yang identified sits right here in the middle, shown here by this tower of triangles. And when we looked at the variants in UK Biobank, on the bottom here, we saw that there was this region that was depleted of variation compared to the rest of the gene in which this variant sat. And this region corresponds to about 18 base pairs. One thing you might note is this gene is incredibly mutation dense. So we actually see about 75% of all possible single nucleotide variants across the gene sequence, compared to a genome average about 13%. So really, really heavily mutated, but depleted in the central region. And when we looked both in Genomics England and also collaborating with others around the world, we very quickly identified other variants and other individuals with variants in this very small 18 base pair region. And actually, it remained that this variant that Union first identified was incredibly significant. So around 70 to 75% of all individuals with variants in this region have that exact same single base insertion. Don't ask me why. I have no idea, and it's really weird. So we collaborated with Anna O'Donnell-Luria's team in Boston and two members of her team, Gabrielle and Sarah, to look at the phenotype involved with variants in this region. So all of these individuals have a severe NDD characterized by global developmental delay and intellectual disability. And the majority of them have microcephaly. They're short in stature. Most of them are non-verbal. They have hypertonia, so low muscle tone, issues with vision and feeding, and a large proportion of them also have seizures. And for any of you that might be dysmorphologists, I'm assuming that's a very small proportion of this room, they apparently have similarities in their faces as well. So what is going on here? So RNU42 encodes the U4 small nuclear RNA, and it's in that trisNERP complex that I mentioned earlier that is directly involved in telling the spliceosome what should be the 5' splice site. So telling it exactly where to cut. This is that trisNERP complex. As I said, it's a complex of both RNAs, shown in color, and proteins shown in gray in the background. And U4 is this red snRNA, which is shown tightly wound to this other snRNA called U6. And it's U6 that actually directly binds to the 5' splice site during its recognition. So to zoom in on the center of that interaction between U4 and U6, here I'm showing you a part of the cryo-EM structure just of this very central portion of the gene, where the bases where we find single nucleotide variants are shown in orange, and the positions where we find single base insertions shown by arrows, this being the primary insertion in the middle here. And what we find is that all of these variants map to two key structures in this duplex. This one here is called STEM3, and this one is called the T-loop or the quasi-pseudoknot structure. But what these two key structures are responsible for is holding in the correct position this flexible linker sequence here, which is the U6 agar-gar sequence, literally sound out the base pairs. And that is the sequence that directly binds to the 5' splice site. So our hypothesis here is that perhaps these variants are disrupting this duplex and in some way disrupting the recognition of U6 of that 5' splice site. So within Genomics England, we had RNA sequencing data for 5 individuals with variants in this region. And lo and behold, we could show that compared to controls, we saw a significant increase in significant events that corresponded to use of non-canonical 5' splice sites. So here where we expected to splice here, it's splicing either up or downstream of that canonical site. Actually, most of the time at weak sites that are used at very low levels in most individuals, we see an elevated use of these non-canonical sites in these individuals with RNU42 variants. These sites are not random. We see the same events in common across these different samples. So here is the most boring histogram in the world, where if we take if we sample 5 controls randomly and look at how often they share the same 5' splice site event as significant, and basically it's always 0, compared to our individuals with RNU42 variants. This is a systematic change in 5' splice site usage. We then decided to look at the sequence motifs surrounding the 5' splice sites that were either increased or decreased in usage in these individuals with RNU42 variants. The ones that are decreased in usage at the bottom here, this is basically just the canonical splice site motif. But hopefully what you can see here is that there are differences at these +3, +4, and +5 positions, where these sites that are increased in usage have an increase in T at +3 and an increase in C at +4 and +5. But what really beautifully joins up the mechanism here is that these exact 3 base pairs of the 5' splice site, +3 and +4 and +5, are what bind directly with this U6 agar-gar sequence, joining up and kind of supporting that mechanism that these variants are acting by disrupting that U6's ability to bind to that 5' splice site. So this was really cool. When we released our paper on this, we had some press coverage, including from The Guardian in the UK, and we had a quote from Nicole Cedar, who's shown here on the right, with her daughter, Mia Joy. And I actually went into a tunnel of realizing things about syndrome naming, and actually how it's not actually that easy. But Nicole actually gave us the name for this new syndrome. So she said that their family liked to refer to RNU42 as renew. as the family is being renewed by this new hope, this information and hope for the future. It's a really clever play on the RNU in that gene name. So that's why we got to renew syndrome. So the thing that still surprises me about this discovery is the prevalence of this disorder that had gone undiscovered for so long. So in Genomics England, we have 60 individuals with variants in that 18 base pair region. It's about 0.7% of the undiagnosed neurodevelopmental disorder patients. Now, it's completely impossible to, from scratch, figure out what the prevalence of the disorder is. And that's because nowhere in the world do we have individuals presenting with NDD, walking into a clinic and immediately getting genome sequencing. That's just not what we do. It's too expensive. So instead, we tried to estimate this using the diagnostic rate from the UK-based Deciphering Developmental Disorders, or DDD cohort, where they had a 40% diagnostic rate in their cohort. So if we assume that the diagnostic rate is 40% before we create our undiagnosed NDD cohort in GEL, that would give us an estimate of about 0.4% of all NDD being caused by variants in this gene. That sounds pretty small, but NDDs are very, very heterogeneous. So let's just flash out the proportions of the 3 key players that contribute the highest number of diagnoses in the DDD cohort. As you can see, these 3 protein coding genes, their percentages are not that much higher. But what's the key difference here is that every single one of these genes is in excess of 2,300 amino acids long. That's 7,000 base pairs of coding sequence for those de novo variants to randomly occur. That's compared to an 18 base pair region of a non-coding RNA. Pretty cool. So given these numbers, we estimate there are around 100,000 individuals worldwide with Renew syndrome. One of them is the wonderful Charlie, who's shown here with his dad, James. And just so happens that James is an editor and writer for the Sunday Times, one of the big tabloid newspapers in the UK. So James wrote an article about his family's story and their journey being undiagnosed and what it means to them to get a diagnosis. For all of us in this room that are working on trying to solve the VUS problem and get diagnoses to patients, please read this article. It's beautiful and it tells you exactly why we're doing what we're doing. But there are a multitude of different outstanding questions that we still don't know. We're really in the early days. It was February last year when Yiyan came to me with this variant. So do variants outside of that critical 18 base pair region cause neurodevelopmental disorders? There's been a series of VUSs that have been proposed, but are they actually pathogenic? Can any in silico tools, variant prediction tools, actually distinguish pathogenic from benign variants? And is there anything special about that kind of crazy single base insertion that keeps recurring? And all of this is somewhat complicated by that really, really high mutation rate that we see across the gene, and also the fact that the whole thing is super highly conserved across evolution, because this is a core splicing factor. Step in some experimental geniuses, at least one of which will be very familiar to you. I went to Greg, I don't know when this was, a while ago now, and said, do you want a new gene to play with? It'll be fun. So Greg and the wonderful Joe and his team did some beautiful functional work on RNU42 for us. It's a slide I left in here, but I don't need to describe this approach to you, and I'm not even going to try because I'm definitely not the right person in this room to do so. But we used one of those essentiality assays in HAP1 to generate a set of function scores for variants across RNU42. So this is what it looks like. So RNU42, as I said, is 145 base pairs long. Beautiful for a small gene, means we can actually fit in the whole gene in a single assay. So we did every possible SNV across the entire length of the gene. We did single base insertions and deletions within the 18 base pair critical region in the middle. And conscious that we might not get enough separation in the assay between the benign and the pathogenic, we also spiked in some variants that we thought might be benign, so some insertions into these loop regions, and also some multinucleotide insertions at the key regions within this 18 base pair region. Just to note that this trying to target this gene is somewhat slightly complicated by the fact there are actually multiple copies of genes that encode U4 across the human genome. So this is just a few of them. This is just showing I think there's something like 90 or so of them, with very high sequence homology. The most difficult one is directly next door on chromosome 12, RNU41, which differs from RNU42 by just 4 different single nucleotide variants, so 97% homology. But RNA42, the reason that we think this is the one driving NDD is this is the expression in brain. It's clearly the kind of the primary brain isoform. But how do we specifically target RNA42 in our SGE experiment? Well, Greg and Joe devised an approach to cut outside the gene where there is sequence degeneracy. So this is what the data looks like. So here, you're very used to looking at these plots, but using those control insertions, we drew this dotted line at a position that corresponds to a score of minus 0.39, saying that everything below this line was significantly depleted in our assay. These scores allow us to perfectly separate out the variants that we find in Renew syndrome. I should say that we don't have many VUSs in this gene, given it was such a new discovery. So here what we're using is the variants that we know are pathogenic, so the ones that are in red found in Renew syndrome, and we're comparing here to variants that are found in the population. So we can see that the ones in red all fall below this dotted line, whereas the ones in the general population, in UK Biobank or all of us, are the vast majority are below this dotted line. And the ones that we don't observe in either, similar to the VUSs, most of them are above the line with quite a few below. If we zoom into that 18 base pair region that is flanked by these two vertical grey lines, we can actually see that we can now further refine this region to two smaller regions of, I think it's 9 and 4 base pairs, where pretty much all of the variants fall below the line, and either in red because they're observed in Renew syndrome, or in teal/green because they're not observed at all. Another thing we can tell is that this primary variant, this single base insertion that we see in most patients, doesn't seem to be an outlier for function score. It doesn't seem like that is an ascertainment issue of it being a more severe disease. These individuals are more likely to present to clinic and get genome sequencing. It seems to be similar to the others. So it still remains a mystery as to why most individuals have that same variant. As I said, there were my numbers. We managed to refine this region to two smaller regions. What we can also look at here is an aspect of phenotypic severity. So what we did is we subsetted the variants in Renew syndrome into two different categories based on an arbitrary threshold of -1, just to kind of split these variants into two equally-ish sized categories. And when we actually use those two categories and plot phenotypic severity, this is a PCA based on scoring each phenotype as severe or not severe. We see that these moderately depleting variants in yellow all cluster to the right-hand side of this plot. That's a little bit obscure, so let's give you some actual solid phenotypes to look at. The one that perhaps works most neatly is if we look at speech. So for the strongly depleting variants, we see that around 70% are completely non-verbal, where this compares to individuals with the moderately depleting variants, and actually over 95% of them can have normal speech or can at least speak simple sentences. So we have evidence that these scores really nicely split out on an axis of phenotypic severity. As I said, we wanted to look at whether whether an in silico tool could work to predict pathogenicity for this gene. This is looking at CAD. As I mentioned, this gene is really, really highly conserved across evolution. And CAD relies really, really heavily on evolutionary conservation in its scores. And you can see that here. On the CAD score axis along the bottom, everything clusters in one big blob. So while there is a significant difference, so with Renew syndrome variants have significantly higher CAD scores than population variants, if you drew a threshold that would classify all of the Renew syndrome variants as pathogenic, you'd also capture 60% of all variants across the entire gene. So basically CAD is pretty useless with an AUC of 0.65 compared to the SGE, which has a really, really high AUC of 0.95. We can also use this to classify the variants of uncertain significance that have so far been identified. So there's a beautiful paper, actually I should have updated this. This came out last week, if anybody wants to look at it. A beautiful follow-up paper from Caroline Naver and Christelle Depienne in Nature Genetics. And they found 4 different VUS variants, 3 of which we capture in our assay, all of which have scores above our threshold of significance, showing that they are likely to be benign. There was also anot' paper that proposed to expand the mutational spectrum out of Renew syndrome outside of that 18 base pair critical region and identify 2 variants down here in this 5' stem loop. Interestingly, both of these variants were inherited from an unaffected mother. I don't know why they called them as pathogenic, well, decided that this meant that they were pathogenic. But anyway, the one variant that we include in our assay is not significantly depleted. So another interesting finding from this, some of you who are eagle-eyed at the start of this might have said, well, actually, you told me the region in the middle of this gene is important. But if you look at the SGE data on that first plot, there are other regions where we see these variants depleting out in the assay. So what on earth is going on here? Well, Greg very quickly hypothesized that this is a haploid cell line, so perhaps these are variants that are recessive. And when you look at these variants, most of them are in navy blue. They're observed in the UK Biobank or all of us. So this supports that hypothesis. This is where they map to on the structure. So here we've got U4 shown here on the bottom here, duplex to U6. And in the middle, these red regions are the critical regions for Renew syndrome. And these other variants map over here, down here, and also up here on this STEM3 region. Sorry, I should have said that here the strongly depleted variants are in red and the moderately depleted ones are shown in yellow. So when we then actually looked in rare disease cohorts across the world, we then found 16 patients from 10 different families with homozygous and compound heterozygous variants that were depleted in our SGE assay that all mapped to these three regions that we identified. Just to take a quick segue before we return to that plot in a minute, there are actually two different spliceosome complexes. There is the major spliceosome, which is responsible for splicing around over 99% of all introns, and that is the spliceosome in which U4 functions. There is also the minor spliceosome, which is responsible for splicing so-called U12 type introns, of which that comprises about 0.5% of all of our introns. And there is an equivalent of U4 with a near identical sequence and structure called U4 attack that functions in the minor spliceosome. Interestingly, U4 attack has been known since about 2011 to be involved in a recessive disease, well, actually 3 different named recessive syndromes for variants that are in U4 attack. And if we plot back on this structure where the variants that are known to cause disease in U4 attack are based, they're in exactly the same regions as these variants that we identified in RNU42, and sometimes the exact same variants. So actually, there's this variant 119A to G in RNU42 with the exact equivalent variant in RNU4 attack is known to be pathogenic in ClinVar. So this is really neat. So we also think that this recessive NDD, although it shares a lot of overlap in being a moderate to severe neurodevelopmental disorder with delayed or absent speech, also has some key differences. And these are kind of towards the bottom of here, is that there is involvement of genitalia and skin and hair abnormalities that we don't often see in Renew syndrome, and also some distinctive white matter abnormalities on MRI. I should also say it's far, far less common than Renew syndrome, with the majority of individuals that we identified being from consanguineous families with related parents. Just to say a bit more about that phenotypic differences. So on the left here, we've compared different phenotypes, whether they're enriched in Renew syndrome that fall to the bottom of this plot, or enriched in this recessive disorder, which fall towards the top. While we've only got small numbers here, not much of this is significant. We see fewer dysmorphic facial features in the recessive syndrome, and we see different abnormalities on MRI, as I mentioned. So while abnormalities of the white matter are not significant here, there are different abnormalities in these two different disorders. So this was the first time I ever looked at MRIs when we got these, so I'm sorry, I'm not an expert. But hopefully these red arrows will point you to the bits that apparently are really, really distinctive. So these kind of dotty white areas at the end of these big X's are periventricular microcystic lesions, which apparently are very distinctive. So distinctive that somebody that we work with in Australia, Kaz Simmons, he said, hang on a minute, this family with biallelic RNU42 variants, I clustered these 10 years ago by their MRI with some other families. One of those families has genome sequencing. Lo and behold, they have biallelic variants in RNU42. So this is so distinctive, we can actually use it to screen and identify individuals to sequence. So what that alerted us to, so one thing I should say is that those individuals that got picked up based on MRI, the variants they have don't actually have significant SGE scores. And when we actually look for biallelic variants across the entire gene, we find a bunch more patients. We now have about 30 patients in this cohort. And they all have variants in the same regions that the SGE identified, but actually some of them shown here in orange. Actually, you cannot see that distinguishing on the screen very well, but some of these variants are shown here in orange. They don't have significant SGE scores, but the variants are in the same regions. And that's in contrast to the variants that we find in UK Biobank. So here we have phased data from 200,000 individuals in the UK Biobank. And you see the vast majority of their variants are found in this middle bit here, which is one of the 3' stem loops. Which has incredibly low SGE scores. So this is a preprint based on the beautiful experimental data from Greg and Joe, so please do go and check it out. There's another one coming focused more on the phenotypic description of this new recessive NDD. So what's been really interesting here is that, well, RNE42 has kind of blown open the field for snRNA research in rare disease, but it wasn't the first. So as I said, RNE4 attack was found in 2011, and RNE12, which is another recessive disorder associated with a minor spliceosomal snRNA, was identified back in 2017. But since we identified RNE42 last year, there's been another couple of major spliceosome snRNAs, RNE22, used to be RNE22P because it was annotated as a pseudogene, but now it's obviously not a pseudogene, and RNE5B1 that have both also been shown to be involved in dominant neurodevelopmental disorders. So it's really, really exciting to see how quickly this field can evolve. So just to conclude, I have no idea where I'm at time because I don't have a clock, so hopefully I'm not too far off. But variants in RNE42, we've shown, of course, both a dominant Renew syndrome and also a novel recessive NDD. And I think this is a really beautiful story to show how powerful SGE is to dissect genotype-phenotype relationships, particularly in a newly identified disease gene. Although we do have that thing we still need to unpick of the fact that these scores are not, while they tell us the right regions for these recessive variants, are not perfectly predictive for this disorder. And the snRNAs are a substantial contributor to previously undiagnosed neurodevelopmental disorders. Just finally to flash up the caveat here, this is a map made by the families. They've already created a non-profit organization called Renew Syndrome United. And on this map, each dot is a family that have a Renew syndrome diagnosis. This is both amazing because there's so many dots on this map and utterly depressing because it shows you where in the Western world we have access to genome sequencing. So we need to do much better to try and get these diagnoses to all patients. And hopefully things like that distinctive MRI phenotype might help us to achieve that. This is some more of the wonderful families. They've got their first conference organized in July. They're absolutely incredible. And I'm looking forward to meeting some of them in New York then. Just to acknowledge, obviously, Greg and Joe for their absolutely beautiful experimental data. Yu Yang for starting this story off. And obviously, I have to say a big thanks to all of the patients, their families, and their clinical teams that recruited them. And this is my wonderful team back in Oxford. Thank you very much for listening.